Assalamu alaikum. Yes, yesterday we discussed about the physiochemical properties in relation to biological action. See, there we discuss ionization, solubility, and partition coefficient. What the role of all these in the biological system? What the role in the human body? Totally in the biological action. What the role of this ionization? Basically, we came to conclude here. These all ionization, solubility, and partition coefficient. They helps to increase the activity. Which activity? Biological activity. Increase the drug action where on the body of the human. Clear? Now the next we will discuss about the hydrogen bonding and protein binding, then chelation. These are also very important, asked many times in our examination. See here, this is the question paper. They were asked many times in our examination. Let's see here. See, role of hydrogen bonding and partition coefficient. Clear? Then phase 1, this is fixed phase 1 type of the metabolism. This is fixed one. In the upcoming classes we will discuss. See, role of hydrogen bonding and protein binding. Fixed question. One is fixed question here for your 5 marks in our examination. Now let's discuss on our present concept, that is on hydrogen bonding. See, very simple here, hydrogen bonding. Here, what is this hydrogen bonding? It is the very weak bond between the electronegative atoms. Okay, now see, this is the hydrogen atom and this is the electronegative atom like the fluorine. This bond, what you are observing, no, that bond is nothing but the hydrogen bond, and this is held by the dipole-dipole interaction between hydrogen and fluorine, dipole-dipole interaction, and the bond is known as the hydrogen bonding. Okay, and when that hydrogen bond will be active, see here, the hydrogen bond, special type dipole-dipole interaction between the hydrogen atom and electronegative atom such as fluorine, chlorine, nitrogen, oxygen, and sulfur. See, this is the water example we have taken. See, this is the H2O, water. Now if you split this water, means HOH. Now see, this bonding what you are observing, no, this one H and H, HO, these bondings is nothing but the hydrogen bonding. Now what's the role of this hydrogen bonding? Why these are very important in medicinal chemistry? We will study in that biological action, or we will study in the uses in here only. So don't quit the video, watch the video until last. When any highly electronegative atoms is attached with hydrogen atom, then hydrogen become electron deficient and they make a weak attraction force and bind with another electron rich molecule. This process is known as hydrogen bonding. What I have explained, no, that only I have wrote here. You can take a note from the description. See, it is represented by the dotted line. This one dotted line, these all are the hydrogen bonds. Mainly hydrogen bonds are two types. One is intermolecular hydrogen bonding, then intramolecular hydrogen bonding. Now what is that inter and intramolecular? See, we will study with an example here. See, my dear brothers and sisters, this is intermolecular, means the bonding of hydrogen bonding where in between the two or more molecules. See, this one is water, this one is also water. Now two molecules of water is joined by which bond? This one is hydrogen bond. Two or more, see, ammonia, these all ammonia is joined by which bond? Hydrogen bond. This is nothing but intermolecular. See, intermolecular is all. Then intra means the hydrogen bond is formed within a molecule. This result in the formation of ring complex. Now see, you have observed here, this one is the benzene ring, and here one more benzene ring is there. In that benzene ring, H and O is joined by which bond? Hydrogen bonding, and they will form a ring here like salicylic acid. Simple. This is inter and intramolecular. Now importance, many physical and chemical properties are affected by hydrogen bonding. Intramolecular bonding, they decreases melting point, boiling point, and solubility of the product. If the molecules are inter, means this type of the molecules inter, then what happens? The melting point, boiling point, and the solubility of the drug increases. It is very important in chemistry and genetic code. Why it is very important? Means, see here, what is this? This is the double helical structure, DNA, you know that. Now see, this ATGC, TAGC, these all nucleic acid, they are joined by which bonds? Hydrogen bonds only. Polynuclear hydrogen bonds only are present where? In your DNA standard chain. So it is very important in the synthesis of the protein and also in the genetic transformation. Huh. Now see, it also increases the water solubility. Yes, of course, it also helps to increase the water solubility. Now our next concept, my dear brothers and sisters, the next concept is of a chelation. See, now what is this chelation? Chelation, it's a very easy, very simple concept. You don't worry at all. Just I will explain you in a very simple way. Chelation, you have studied this chelation in your analysis, pharmaceutical analysis, where in the first semester. Chelation is nothing but formation of the complex. If you know this one means, definitely you will clear your concept here. See, this one is a metal ion, this is the ligand. When metal ion and ligand, they both combine means they will form the complex. This formation of the complex is known as chelation. Simple. Ligand means these are electron donating groups, means they are rich in electrons, they are rich in the electrons, so they are having the property to donate the electrons. So when metal ion made to react with the ligand, they will form a complex. That complex is known as metal ligand chelation or metal ligand complex. Let's see that chelates are the complexes of the metal and ions which are formed by the metal ion and an electron donating molecule known as ligand. Metal ion must have incomplete valence shells, means they should be incomplete valency, outermost electronic configuration. Okay, they should be incomplete in nature. Then only this type of the complexes will be formed. Now see, this one is the example: EDTA, ammonia, NH2, NH2, CH2, CH2. Now this metal ion and which one, this ammonia, they both combines and they will form a ring type structure, that is known as chelation, ring type structure. Now what the use of this chelation in pharmaceutical industry or in medicinal chemistry means, generally chelates form ring structure and disodium EDTA is one of the common important. See, one of the very important here I want to tell you. See what happened, my dear brothers and sisters, lead. Now this is lead is the poison, metal poison. Now sometimes what happen, if a person consumes lead means immediately he will given by EDTA. EDTA is a complexing agent. Now this EDTA, what it will do, it will combines with this lead and form a complex. This complex, which one? EDTA lead complex, easily excreted through urine. Okay, through urine, it can easily excreted through urine. Okay, if lead is present in your stomach means it causes cancer or various reactions can be faced by your body. But if we give the EDTA complexing agent means we can stop this one. Clear? Now this one is the main importance here to conduct the chelation. Disodium EDTA form chelates with the various toxic metal ions and helps in their excretion. Then lead and vanadium poisoning, hemoglobin and cyanoglobin, they are natural occurring chelates. Dimercaprol is chelating agent used in the treatment of arsenic poison. Okay, if any person consume the arsenic type of the heavy metal means we are giving him a dimercaprol. Dimercaprol can be available in the injectable form also. Okay, dimercaprol, it is a complexing agent. It will combines with this arsenic and form a complex. There is no chance of reactivity in the human body. Clear? Now my dear brothers and sisters, our last concept here is a protein binding. Very important one. Protein binding, very important. See, protein binding, simple. What here? Drug binds with the protein and there will be formation of the drug and protein binding. Okay, it is the process in which drug molecules bind with the protein. It forms a complex type. That phenomenon is known as protein binding. Simple. See, this is the complex form. This one is the protein. This one is the drug. These two complexes is known as complex form. Now what the role of that protein binding? Why it is very essential in the medicinal chemistry? We will discuss in the biological action. See, after the absorption of the drug. See, before going to that, I want to explain you. See, my dear brothers and sisters. Now your right hand starts paining. Assume that it is your right hand. It starts pain. Why? Due to some of the reason. Okay, now what happens if pain occurs means you will take one tablet like aceclofenac. Now that aceclofenac tablets, what it will do, first it will enters into the stomach, there it absorption takes place, then it enters into the intestine, then through the help of the intestinal cells absorption takes place. Okay, after that the drug enters into the circulatory system in your blood. Then what happens, my dear brothers and sisters, to increase the activity, it combines with the protein. like albumin or anyone. Later we will discuss one reversible protein, irreversible protein. Just remember, if your drug get reacts with the protein, means they form the drug protein interaction. Now what happens, they revolves all in our, or they move in our circulatory system. Now suddenly these drugs, they will interact with the receptor cells. Receptor cell, these are the receptor cells. Okay, now after the drug, when this interact with the receptor cells, automatically the pain or your swelling get decreases. Okay, how? Why? Because of this drug acted on the receptor cells and they decreases the painness. Okay, they decreases the rashes there. This is the property of the drug, very simple property. Now you understand the concept. See, some proteins, many proteins are there, like albumin, alpha acid glycoprotein, lipoprotein, then globulins. Now there are different types here. Mainly two types of complexes are formed in our body. One is reversible and irreversible. Irreversible, here the drug is bind with the protein with very weak attraction forces like van der Waals and hydrogen bonding, so they can easily detach as the drug, become free, and this drug bind with the receptor and gives a pharmacological action. See, I have told you what happens when the drug consumed. No, it starts the absorption in the intestinal cells with the help of the intestinal cells. Now that drug binds with the protein. See, after the finding of these receptor cells, these protein and drug, see, this one is the protein and drug, they are attached by the weak van der Waals forces. When the receptor is formed, no, these will break down here. Okay, why? Because of weak van der Waals force and weak hydrogen bonding, these dipole-dipole interactions break down and the drug acts on the receptor cell. This is known as the reversible. Now irreversible means just opposite here. See, protein and drug, they are attached by the bond known as the covalent bond. Now you know that covalent bond, these are the strong bond, these are the receptors, means infected site or affected site. Now these, due to the not breaking of this bond, what happens, there will be no activity on the relation, no activity where on the receptor cells or the infected part. No activity means no cureness, so waste of that thing. But in some conditions, irreversible also works and reversible also works. Now effects of protein binding on biological system, main here is absorption. Okay, absorption is very important. It helps in absorption of the drug as it occurs by transfer of drug from the site of administration into systemic circulation. Very important thing here is sometimes what happens, this drug, due to its low stability, what happens, it get degrades here, where in the walls of the stomach or in the stomach only, or in the intestine, or anywhere in the circulatory system, it get degrades, it won't reaches to the receptor cells. But with the help of this protein, what happens, it held with the drug here. Protein, what it will do, it holds this drug, it holds with drug with very responsibility. It won't leave the drug when until it reaches to the receptor. If it reaches to the receptor, means this protein will get separated and drug acts on the particular receptor cell and reduces the pain or infected part. Clear? Yes, my dear brothers and sisters, these are the very important concept, and don't worry about the notes. You will get the notes in the description. And these are very important for 5 marks and also for 2 marks. Clear? This type of the videos you will get, inshallah. So subscribe my channel, Let's Join Pharmacy. And the next important concept, inshallah, we will discuss tomorrow, that is the bioisoterism. It is also very important. Thank you. Assalamu alaikum.